Hi, let's continue on the transport phenomena in biological systems. So today we want to discuss about the relative importance between diffusion and convection depending on the length scales. A lot of these materials are from transport phenomena in biological systems by Trotsky. So I brought this table about relative length scales in biological systems. So we know a lot of these entities, such as proteins and nucleic acids, that length scale is about, I like to put it as a 10 to the minus like n of meter. So that's, we know that the size of the protein is relatively about 10 nanometer order, and organelles are about 10 times higher. And like the cells composing our body for eukaryotic tissue is about, about 10 micron length scale, so which is 10 to the minus fifth length. So that's about 10 micron. And interestingly, the capillary, which is the business end of our oxygen and nutrient and waste exchange at the tissue, is about 10 to the minus fourth meter. And that turns out to be smaller than about 100 micrometer. And so that has an implication today we will discuss. And organ size for human is, let's say, about like 10 centimeter. If it's a kind of big organ, then that's a 10 to the minus first. And our whole body is an order of meter scale. So if you look at the our an organismic body to the smallest machinery, protein, it's like covering a huge 8 orders of magnitudes. And that length scale is an enormous layers of coverage. So what that means is no single transport processes can function effectively over these many length scales. So what we want to discuss here is at shorter distance, yes, diffusion can be quite rapid, while a longer distance, convection typically transport molecules across. So that's we want to argue this time. And what we previously discussed a lot about diffusion and random walks, this is a kind of a hypothetical mathematical description of how we can analyze diffusion process more analytically and easily. So the conclusion from there is diffusion time increases as the square of distance. So what that means is, hey, diffusion time, if the length scale is, let's say, twice, so we want to diffuse twice, then the time for diffusion will be 4 times. If the length scale is, let's say, 10 times longer, and the time for diffusion is simply 10 square, which is 100 times. So as the length scale or the diffusing distance gets bigger and bigger, the diffusion alone becomes a pretty inefficient process. So here I brought this table from the Trotsky book, and here comes some table of diffusion coefficients. So mass diffusion. So for example, here small molecules and gases, those have about 10 to the minus fifth here centimeter square per second, while a proteins inside the liquid has about 10 to the minus seventh order centimeter square per second. So that's something you want to keep in mind. And so we are discussing diffusion versus convection. So in French physicists called Jean-Claude Eugene Becquelet already kind of defined this ratio. Likewise, we defined previously Reynolds number, which is an inertial force versus viscous force. So likewise, Becquelet defined as a convection, mass transport by convection versus mass transport by diffusion. And it's pretty simple. And you can see here, it's so that's mass transport probably is inversely by the time scale. So it becomes a diffusion time divided by convection time. And how can you define diffusion time? Basically, diffusion coefficient unit. If you look at the unit, it's length square over time. So that will be the time will be length square divided by diffusion coefficient like this. And then how can you come up with a convection time? So convection is a simple displacement by velocity or mass flow. So that's pretty much the time for convection will be distance divided by characteristic velocity. So that's L over V. And you calculate this and it turns out velocity times length over diffusion coefficient. So what this means is here L is a characteristic length and V is this local flow velocity, and D is a diffusion coefficient or mass diffusion coefficient. So if Pecklin number is a very big, that means this is a convection dominant, okay? And on the other hand, it's very small, then we can consider diffusion dominant process. So I want to change into a board exercise to discuss this in more mathematical point of view. So let's start some board exercise. The topic is comparison between diffusion and convection. So first, let's define the diffusion time. Diffusion time as T sub diff can be defined as, think about diffusion coefficient unit, it's length square over time. So this must be length square over diffusion coefficient. Let's put this as a equation number 1. Likewise, convection time. Convection time as T sub convec. And that will be distance over velocity. So, okay, so that's distance over velocity. So then what are we interested? We are interested in these two times crossing over or matching together. So when T diffusion equals T convection, then when does that happen? That means, let's say this length as a characteristic length as L star will be defined as D over V. And diffusion coefficient, as we saw it, 10 to the minus n. So I'll just write down in this way, 10 to the minus n and centimeter square over second. While velocity or local flow velocity, let's just, yeah, we can set up as a, for example, 1 micrometer per second. Just let's assume. Okay, then let's, to make these, to compare these, like there's a L to the square, so it's power. So it's sometimes easier to consider and compare this in log scale. So let's just take a log of these. So let's take ln of 4, 1, and 2. Then what we will get is ln T diff, diffusion coefficient, with 2 times ln L minus ln diffusion. And like ln T convec will be ln L minus ln V. Let's make everything as a non-dimensional. So how can we draw this? So that as a number 3, number 4. So this will be x-axis as the ln length scale and y-axis as ln t. So then the slope for this convection will be 1. Let's say slope of this is convection. Then what is the slope of diffusion, which is 2. So we will be having bigger growth of this. Let's write it this way. Okay, so that's diffusion. All right, so what does this mean? There will be the crossing point, and that we can define as ln L star, which is when these two becomes the same. And we can now understand this from here. So that on the right side is a, now thinking about time is a faster means more efficient. So on the right hand side is convection time is smaller. So it's convection dominant region. While this left side is diffusion dominant. So what does this imply? Is depending on the characteristic length. So longer scale, convection will take less time, meaning that convection will be more efficient. On the other hand, it's a smaller scale than L star, then diffusion will take less time or diffusion process will be more efficient. So this gives an insight of how and why our organisms has developed from bacteria to eukaryotic cells and multicellular organism and like primates like us, which develops an efficient circulating system. So let's have an example. Let's, for example, for let's say proteins, which has about diffusion coefficient, let's say D sub P is about 10 to the minus 7 centimeter per square, then we can compute this L star, assuming like, let's put the velocity at 1 micron, then what it'll be like 10 to the minus 7 centimeter square per second divided 1 micrometer per second, which turns out to be about 10 micrometer. And okay, so that's good number to consider. What about small molecules or gases? So that's diffusion of, let's say, gases is about 10 to the minus 5th centimeter square per second. Let's go back to check the diffusion coefficient of small molecules in liquid, 10 to the minus 5th. Then let's see, L star, the 10 to the minus 5th centimeter square per second. Let's see, about this is about 100 times longer. Let's put as a length scale of 10 micrometer per second. What will happen over here? So centimeter, 1 centimeter is a 10 millimeter, and 1 millimeter is 10 to the 3rd micrometer. So that's the 10 to the 4th micrometer, about that. So this will be 10 to the minus 5th, 10 to the 4th square micrometer square per second over 10 to the micrometer per second. And this will be 832. So about 100 micrometer per second in this case. So let's see. I just note that this is just a simple approximation, just to try to understand the implication of this. What does that mean? So protein case, this length scale for characteristic length is smaller, that we kind of understand. So So what this means is a very small scale, like length scale is a 10 micrometer or even small, a cellular size, then if, this is kind of characteristic length, or smaller length scale, diffusion will be dominant for that. And this is the crossing point. While let's say gas or small molecules, if we assume that the local flow velocity is 10 micrometer per second, then what comes out is 100 micrometer per second. And that's kind of, the length scale smaller or bigger, and these gases will diffuse more efficiently by diffusion if the length scale is within 100 micrometer. So I hope this one gives you some insight for understanding this phenomenon. Okay, I want to add a little bit on here, because when I look at the table, proteins in liquids can have diffusion coefficient 10 to the minus 7 till 10 to the minus 6, which is 10 times bigger. I want to change velocity as a 10 micrometer per second, and that will have 10 to the minus 6 to this will be about 1 to 10 micrometer. So for proteins to move around, diffusion will be efficient in 1 to 10 micron scale or less. So which means probably inside the cell or bacterium, which has the size of 1 micrometer, diffusion alone will be enough, and we don't need any convection or relevant. While gases and small molecules, the diffusion coefficient, the versus convection, this crossing over happens about 100 micron. Okay, so I think I have a wrong unit here. So about 100 micron. So with smaller than 100 micron, we probably don't need a vessels because diffusion is still efficient enough. So hopefully this gives you a good idea and insight. So here's the kind of summation, summary for that relative importance of convection and diffusion. So this lower slope is talking about convection, while higher slope is discussing about convection, diffusion process. So with respect to different two diffusion coefficient, there's like two crossing over. So crossover happens around, let's say, gases in liquids around 100 micrometer, while proteins in liquids, it's around 10 micrometer and this order. But it depends on how we set up, assume like the characteristic velocity. But the important aspects from this kind of analysis is for proteins, diffusion is efficient, dependent of the size of cells or smaller, which means, like inside a bacterium or a cell, proteins can be very efficiently diffused so that it can cause all this metabolic activity, enzyme activity inside the cell. But for small molecules such as gases and glucose, and diffusion is efficient over the distance of like about 10 times bigger or 100 micron. So that means is these gases can diffuse at least tens of micrometer. So so we have I found a paper in PNAS that what's the normal spacing between capillaries in many tissues. What this reference tells us is generally to less than 50 or 100 micron is capillary spacing. So that means, inside the tissue, which require this gas, oxygen and CO2, like, or this exchange will be efficient enough with the diffusion, so that our convection convection at the end will be based on this capillary. So the capillary to capillary, the distance will not be bigger than 50 or 100 micron. So that gives us how the nature, when we develop our system for effective convective transport of molecules, and if at the end, out of this capillary, that shorter distance diffusion is enough, or inside the cell, the diffusion process is fast and efficient enough for this or exchange of molecules. So with this, next time we will discuss about transport across cell membranes among cells. Thank you for your attention.